Welcome to the 81st Annual Cold Spring Harbor Laboratory Symposium on Quantitative Biology. This year's topic is on targeting cancer. I'm Mirna Kvajo from CSHL, and I'm sitting here today with Sean Morrison from UT Southwestern Medical Center. Hi, Sean.
Hey, how are you?
So, Sean, you're studying stem cells, and I know that's a very, very broad statement because stem cells, by now are being studied in so many contexts and so many questions are being asked. And I think, but I also know that you're actually interested in one specific process that goes along with stem cells, and that's the process of self-renewal.
Mm-hmm.
That's the ability of stem cells to really, create more stem cells, and in this way allow them to kind of support the regenerative and developmental processes. And I heard on this conference that people think that self-renewal is really something that distinguishes stem cells from the rest of the cells. So can you tell me a little bit more about this very important process, and actually, why did you decide to study it?
Self-renewal is the process by which stem cells divide to make more stem cells, and it's fundamental to their ability to persist throughout life in your tissues. So we have to have stem cells that persist throughout life in your skin to make new skin cells, in bone marrow to make new blood cells, in bone marrow to make new bone cells. There's many different kinds of stem cells. And when I first started my lab, now 17 years ago, and I was trying to think, what is the most important question in stem cell biology? I thought it was to understand the molecular mechanisms that regulate stem cell self-renewal. Partly because stem cell self-renewal is necessary for the expansion of stem cells during development, for the formation of tissues. Partly because it's necessary for the maintenance of stem cells throughout adult life to maintain our tissues. Mm-hmm. And there was also a sense at the time that it might have something to do with cancer. And at the time, it seems funny in retrospect. At the time, people were thinking, well, there's pro-proliferation genes and there's anti-proliferation genes, and so of course, stem cells are going to depend on pro-proliferation genes. Mm-hmm. It turns out to be a lot more interesting than that. And one of the things that we learned as we started studying stem cell self-renewal was that self-renewal is regulated by networks of proto-oncogenes and tumor suppressors. And we were bumping into the same genes that regulate cancer cell proliferation. But it's not as boring as there just being pro-proliferation genes and anti-proliferation genes, because one of the things that we learned is that stem cell self-renewal is different from the proliferation of restricted progenitors in the same tissues. And so we went back and forth between hematopoietic stem cells and neural stem cells, trying to identify key regulators of stem cell self-renewal, which we define as gene products that are required for the self-renewal of the stem cells, but which are not necessarily required for the proliferation of restricted progenitors in the same tissue.
And so you're now talking about different types of tissue. Do different types of tissue also require different regulators of stem? Is that
obviously there are different regulators, but what was more remarkable is that there's a group of regulators that are incredibly conserved across stem cells in many different adult tissues. And those tend to be the genes that get hijacked by cancer cells. And so when we would run into a new regulator of stem cell self-renewal, invariably it would turn out to be involved in cancer later. And so what that means is that, at least to a first approximation, it looks like cancer cells hijack stem cell self-renewal mechanisms more so than restricted progenitor proliferation mechanisms. Presumably there's some advantage to cancer cells to hijacking molecular mechanisms that enable a long-term capacity to replicate themselves rather than the limited replicative capacity that typically you see in restricted progenitors.
Could you speculate what could be the advantage? What is so special about this self-renewal process that could be so advantageous?
I think what it is just that cancer cells need to replicate themselves extensively. Stem cells are distinguished from restricted progenitors by being able to replicate themselves extensively, whereas restricted progenitors often have a limited replicative capacity. And so it makes sense from that point of view, but what it means is that cancer is a disease of dysregulated self-renewal. And the mechanisms that we think of as being so important in cancer biology, they didn't evolve to regulate the proliferation of cancer cells. These networks of proto-oncogenes and tumor suppressors evolved to regulate normal tissue homeostasis. And their role in cancer reflects the ability of cancer cells to hijack those processes to enable a selfish neoplastic proliferation.
And do you think that this this is really a mechanism that is very general for all of the cancers?
Many cancers,
Many
probably
cancers.
not all cancers. You know, for example, you see some mechanisms in T acute lymphoblastic leukemia that are borrowed from restricted T lineage progenitors, as Ionis Siphontis and others have shown. Now, T cells are more like stem cells, though, in the sense that they can go in and out of the cycle for long periods of time and persist throughout life. So in a sense, they're restricted progenitors, but on the other hand, their mode of division is a little bit more like stem cells.
So you also said you are kind of looking for new regulators all the time, right? You have this core of conserved regulators, but then you also look out and try to find other regulators. And I also know, I think from your talk and you know your studies that you really look at regulators which are also somehow something that maybe we think about regulating kind of more general systems. You know, I mean, I can give an example, for instance, of estrogen, I guess, and leptin, right? You kind of implicated all these molecules in stem cell renewal, which is really interesting because we think of them as something that kind of maybe affects our whole body in a myriad of ways. So
Yeah.
How do you think of that?
Well, you know, when we first started studying self-renewal mechanisms, there was almost nothing known at a molecular level. And so when we would identify a new transcription factor like SOC17 that was required for hematopoietic stem cell self-renewal, or an epigenetic regulator like BMI1, it was a Nature paper or a Cell paper. And now the kinds of approaches that we were taking to identify genes that were necessary for stem cell self-renewal, there's a lot of people now who are taking similar approaches to look for transcription factors and epigenetic regulators and signaling molecules. And so, you know, we think a lot about the kinds of experiments we do. And there's some kinds of experiments where the question is obvious and the way to answer it is obvious. And so we could compete, you know, to be the first one, but whether we're first or whether we're second, there's no question that is going to get answered by the field. And so whether we do it or not, it's not really going to change the way the field develops. So we're more interested now in studying aspects of cellular physiology that haven't been studied before in somatic stem cells.
Yeah.
And often, you know, generally they haven't been studied before because people are used to using 10 million cells, and you can't get 10 million cells if you're studying somatic stem cells. So we end up having to scale down assays, and as a consequence, you know, we're studying proteostasis in stem cells, we're studying metabolism, all trying to address the general question of whether stem cells differ from other cells based on based purely on executive control, where there's a few, you know, transcription factors or epigenetic regulators that distinguish stem cells from other cells, but maybe a lot of the other cellular physiology is the same. You know, maybe the way you segregate your chromosomes, the way you metabolize glucose, the way, you know, maybe most of that is basically the same as in many other cell types. The other general possibility is that in order to create a stable, self-replicating, multipotent state, that there's many aspects of cellular physiology that have to be different in a stem cell. So that's what we're trying to get at, because we think that there's an enormous amount of new biology that can be understood by understanding how things that we at some level think of as housekeeping functions, whether it's metabolism or protein synthesis or whatever, how it's done differently in different kinds of dividing somatic cells.
You also seem to be interested in the context in which the stem cells live, right? I mean, because stem cells live in their own little worlds, right, which promote their functions, and that's a very hot topic of study, because people really started thinking in a very holistic way about questions like, okay, how does the environment affect this and that? And for stem cells, this is also the case. Can you tell me a little bit, you know, like what's your take on the importance of specific environments for the stem cells function?
Stem cells, we think, do depend on highly specialized microenvironments that we refer to as niches for their maintenance. And so, yes, one of the major interests in my laboratory is to understand both local signals from the niche that maintain stem cells, particularly blood-forming stem cells, as well as long-range signals. You mentioned estrogen and leptin before. I think one of the fundamental questions is to understand, you know, we must have mechanisms that integrate stem cell function across different tissues, right? When you have a full-body physiological change like starvation or pregnancy, stem cells in all different tissues have to be reading from the same song sheet. You can't have stem cells kind of making it up as they go along in different tissues. So there have to be mechanisms that integrate stem cell function across different tissues with full body physiological changes. And estrogen and leptin are two examples of factors that do it, at least in certain stem cell systems. And we're trying to better understand the local microenvironment that maintains stem cells. Most people don't realize that a lot of the growth factors that we know of and that are used in regenerative medicine are either so highly and broadly expressed that you can purify them from urine, or that they're members of a big growth factor family so that they can be identified by homology with other family members, BMPs, WNTs, TGF-betas, Notch ligands, etc. And yet, even though we've identified scores of growth factors from these families and that can be purified from urine, there are certain things that we still can't control in terms of regenerative medicine. We still can't really grow hematopoietic stem cells in culture, and that is limiting for our ability to do bone marrow transplants in a lot of patients. And we still only have a single anabolic growth factor that's FDA approved to promote the formation of new bone, and osteoporosis is a huge unmet medical need. So we're looking in the bone marrow to try to see whether there are additional growth factors that haven't been identified yet that are highly restricted in their expression to specialized microenvironments like niches, and that explains some of that biology that we can't control right now. And we do think there are growth factors. We're in the process of characterizing some of them now, and hopefully some of them will be useful in the context of regenerative medicine.
People sometimes talk also about tumorigenic niche or tumor niche. Do you think, when you think of stem cells, stem cells as kind of potentially source of tumors, do you also think that they also need a right environment to create the tumor? I mean, is this something that you're thinking about?
It is something that we think about, because we go back and forth between trying to understand mechanisms that regulate normal stem cell self-renewal versus the self-replication of cancer cells. And on one hand, of course, cancer cells are influenced by their environment in lots of different ways. And of course, that can have important consequences on which cancer cells are actually fated to expand and which cells are not, and which cancer cells are therapy resistant. But on the other hand, I think it's important to keep in mind that cancer cells are less factor dependent than normal stem cells. And so people talk about niches for cancer stem cells, and I'm sure that there will be examples of that. There are already some compelling data. But I think for the most part, cancer cells aren't going to show the same obligate dependence as normal stem cells on highly specialized microenvironments.
They're
because
robust.
In the end, right, we know that they're invasive and that they overgrow tissues. And when you get a breast cancer cell or a melanoma cell or a prostate cancer cell in the bone marrow, they don't only grow in the restricted microenvironments where normal stem cells grow. Eventually, they ablate the structure of the tissue and they grow everywhere.
I see.
So it's clearly worthwhile to study the microenvironmental mechanisms that regulate cancer cells, but it'll probably be more of a facultative dependence or a situational dependence than what we see with normal stem cells.
So I think the stem cell field, as you said, has been, as most fields, are somewhat limited by the technologies, right? What you can do, especially if you think about studying stem cells in vivo. That's such a formidable challenge. There aren't that many of them, and the way you can actually look at what's going on, it's really challenging. And I think you mentioned that you've been looking at metabolic processes. Again, this is something that we can currently study in relatively large numbers of cells, so there's a need to scale down. So I'm thinking, when you think about technology and when you think about what needs to happen to kind of move, say, your research or in general the stem cell research to the next level, is there like do you have a wish list? Do you wish for something to happen or for someone to come up with something specific that would make your life easier?
Be nice to have a PCR for proteins.
A PCR for proteins. Okay.
Yeah, that would change the world if we could PCR amplify proteins and not just genes.
Okay. Well, that's a very interesting idea. Whoever's watching this, so
that's number one on my wish list. Beyond that, it's a technique by technique. Throughout my entire career, we've always had to scale down every technique that we work with. And the thing is, it's usually not hard. Usually people use 10 million cells because they have 10 million cells, not because they need 10 million cells. And if you're just a little bit more careful, it's amazing how easy it has been to scale down a lot of things.
So you don't take things for granted when it comes to technique, you really try out.
We always just assume that we're going to be able to scale it down, and sometimes it's easy and sometimes it's more challenging. You know, right now we want to study metabolism in small numbers of highly purified cells and really understand how metabolic pathways are used differently by different kinds of dividing somatic cells in the same tissue. Collaborated with Ralph de Berardinis in our institute at UT Southwestern to scale down metabolomics. And we're doing metabolomics in 10,000 cells and discovering new pathways that regulate stem cell function.
Great. Well, on that note, I should say that's such an exciting research, and thank you so much for taking the time to talk with me, and I wish you all the best in your research and look forward to reading more.
Great talking to you.
Great. Thanks.